Good morning, Dr. Murphy.
Good morning. Good morning, and thank you for inviting me.
Thank you. So today's guest is Dr. Brian Murphy, who is the CEO of Emerald Bioscience. Dr. Murphy, I've looked at your background. You only have an MD and a MPH and an MS or an MBA, and there's a couple of Ivy League schools that are in the background. So I feel like you got to tell us why so many degrees and why such a interesting career. You've done quite a bit.
Well, I'm from New York City originally, and I did my medical school training in New York. I originally was intending on doing a combined MD PhD program in research. I ended up taking a course in epidemiology, and from there, I decided to change focus. And so I ultimately earned my MD degree and then a Master of Public Health in epidemiology. And then I had done a lot of basic science work in parallel to that. So at that time, I also earned a Master of Science in Pharmacology, which helped serve as a good basis for my interest in drug research later on in my career. And my MBA, when I completed my fellowship, I did combined fellowship tract studies at Harvard Medical School and the Harvard School of Public Health. I taught at both of those institutions. When I returned, when I left Boston and came back to New York to practice, this was a time of upheaval in the medical profession as far as practices and the introduction of managed care. And so at that time, I was accepted and completed my studies for a Master of Business Administration at Columbia University. And in addition, after earning my MBA, I was on visiting faculty there for a bit related to mentoring and being involved with students that were interested in entrepreneurship.
Mhm.
So that's kind of like the 30,000-foot view of all of that.
Fantastic. So it sounds like you have a true passion for science, particularly epidemiology and perhaps drug development. At what point did you, at what point, or how long did you practice, and at what point did you decide to make that transition from practice into the pharmaceutical industry?
I left practice circa 2001. I practiced for more than a decade, and that included seeing patients, a wide variety of patients. I had a practice in the Wall Street area of New York, and I also had a practice in a part of New York called Hell's Kitchen,
Oh, wow.
which served populations that were medically underserved and was involved with a lot of charity work, which actually was very rewarding. I had also worked. Practiced in the VA system as well when I was in Boston. And in fact, I did my chief residency. I was a chief resident in internal medicine in the Boston University system. And I did my chief residency year and a couple of years after that, when I was a fellow, I continued to practice at the VA. Ultimately, I was practicing, I was teaching medicine, clinical medicine, and also performing research predominantly in infectious diseases. And as the saying goes, life is what happens while you're making other plans. I was giving a medical grand rounds in New York City at Mount Sinai Hospital on drug development and hepatitis C therapy. Unbeknownst to me, the CEO of Hoffman LaRoche was in the audience. he came up afterwards and said that they would be interested in recruiting academic physicians into their programs, and ultimately it was at that point that I made the leap from physician academic into physician industry physician within the pharmaceutical industry. So my first job in the pharmaceutical industry was as medical director of North America for Hoffman LaRoche in the areas of anti-infectives, including antibiotics and antivirals, as well as transplants.
Amazing story. Now, I'll backtrack a little bit. When you were a practicing physician, I'm just wondering in the VA, did you have a lot of patients that were chronic pain patients and in either using some kind of a substance to alleviate that pain or being prescribed it? And I'm just wondering if you've had that encounter, how you feel like that method of treating those patients has evolved since cannabis has
Well,
become legal.
that's a great question. During my practice time, as I mentioned, while I was boarded in internal medicine, I did subspecialty training in infectious diseases as well. So a significant proportion of my patients were HIV positive, and it was at a time when there weren't a lot of treatment options for those patients. And one of the drugs we were, my practice group was involved in studies of a drug called Marinol, which was the first approved therapy of a cannabinoid. The cannabinoid was THC, and it was an oral form of THC. And it was developed, and ultimately the FDA approved use was for management of chemotherapy-induced nausea and vomiting. And you could look at some of the therapies used, the antiviral therapies at that time for HIV could be considered chemotherapy. And the secondary use that was approved was for a wasting syndrome related to HIV infection. So in addition, I also had patients that used cannabis during that time to help alleviate some of the side effects associated with the very early therapies that were being developed for HIV, which had a lot of, I would say, gastrointestinal adverse events, just like chemotherapy. Currently, a lot of chemotherapy for used in oncology for cancer chemotherapy. A lot of those therapies also come with adverse events associated with lack of appetite, inability to keep food down, inability to control nausea. And so the cannabinoids as a drug and cannabinoids derived from the plant at that time proved to be, as they are today, have proved very useful in helping patients tolerate therapies.
So, what was the common, I guess, outlook of a physician at that time? Were they open to patients using cannabis on the side for monitoring these side effects, or was there a negative bias toward the plant at that time?
So you're looking at a time in the, especially as these therapies were first introduced, you're looking at the late 80s through the 90s, and it was very hard to be able to tease out whether or not these cannabinoid therapies were effective because one, having a viral infection like HIV and all the other opportunistic infections associated with that sometimes counteracted the ability to show a drug benefit. And then also at that time, THC, then as it is now, still considered a Schedule I controlled substance. And so at that time, I would say that there was still a stigma associated with the plant, which I would say is still out there to some extent today, but certainly not as bad as it was 1 to 2 decades ago. I think things are getting a little bit better, and we're turning the corners related to the stigma associated with cannabis as far as the plant is concerned, and cannabinoids, the actual molecules derived from the plant.
Well, thank you for your work as one of the early adopters in the medical establishment. I think your ability to understand some of the science and be able to translate it to some of us is really instrumental in pushing the movement forward. So I want to say thank you for all the work that you do. So tell
Well,
us
thank
how you got involved with a cannabis research company.
Well, one of my former jobs, I served as a chief medical officer of a company called Valeant Pharmaceuticals, which is also located in Southern California. That company has since undergone a transition and a name change. It's currently called Bausch and Lomb, but at the time was called Valeant Pharmaceuticals, and that company had in-licensed, and we ended up relaunching in 2005, 2006, the second approved cannabinoid therapeutic. The generic name is called Navalone. The trade name is called Sesamet. This is another oral form of THC, like Marinol, and has the same approved indications for use in chemotherapy-induced nausea and vomiting, as well as AIDS wasting. So between doing research in the past with Marinol, and then by that time, 2005, 2006, I was in on the industry side, having helped relaunch that drug. I was pretty familiar with the potential of cannabinoids and cannabinoid-based medicines as far as impacting health. It from a medical pharmaceutical perspective, early on, probably in the 80s, 70s, and 80s, it was recognized that probably it was thought of, at least at that time, that there were only two organs that possessed receptors for cannabinoids. And it was thought that the brain only had cannabinoid receptor type 1, and the bone marrow only had cannabinoid receptor type 2. Since that time, medical research has gone further, and we now know that cannabinoid receptors are located in just about every major organ in the body, and that those organs can contain not just one distinct cannabinoid receptor, but can have both types of cannabinoid receptors. And even in the brain, you can find cannabinoid type 1 and 2 receptors, and the same story for the bone marrow and the immune system. So our understanding of just the far-reaching effect. If the audience was very scientifically focused, the word most associated with cannabinoid medicine and their receptors, the word most associated with that is what we would call a pleiotropic response, which is a fancy scientific term that these drugs can do a lot of things, basically, in a lot of different organs for a lot of different indications. And so this is the state of cannabinoid, at least from the pharmaceutical perspective. This is where we are right now, gaining a better understanding of the use of these drugs. And in particular for this company where I serve as the chief executive officer, Emerald Bioscience, we are focused on honing that development, that drug development, even more to a precision medicine perspective, whereby using, and we call them derivatives of cannabinoids. So these a derivative can be a prodrug or an analog, and I can go into a little bit of more detail of what that means. But by using a derivative of a cannabinoid, we are able, our quest is to deliver the right cannabinoid in the right concentration to the right target organ at the right time, so that we can be, it's almost like a surgical strike of getting the right drug to the right organ for the right disease. And this way, we do this in the hopes of optimizing both safety and efficacy for these drugs.
Amazing. So some of your current research right now has to do with work on glaucoma. Can you
Correct.
tell us a little bit more about that, and why would an average person care about that?
Well, going back a little bit to the statement I just made, we have a partnership with the University of Mississippi, and we have in-licensed uh, all intellectual property related to THC and CBD from the university. What's important about the University of Mississippi is that they currently still hold, or the only entity that holds a federal license to grow, cultivate, and research cannabis autonomously. And they've held that license since 1968. So for more than 50 years, they have a tremendous amount of intellectual capital related to both the chemistry and physiology of cannabis and cannabinoids, the molecules derived from the plant. As I just mentioned, we are working with derivatives. So what I'd like to do maybe is just to give your audience an idea of what a prodrug is as opposed to an analog, and how that relates to glaucoma, if I may.
Absolutely, that would be amazing.
So a prodrug is a modification of an active compound to make that compound more amenable to entry into the human body. And so we have a prodrug of THC that we are developing. It's our lead compound for the treatment of glaucoma. What is interesting is that we know already, based on studies done by the United States Army and the NIH, that when you smoke marijuana, you can correlate the effect of THC on lowering intraocular pressure in the eye. And elevated intraocular pressure correlates to damage of the optic nerve, which is what happens in glaucoma. So the NIH, when they studied patients smoking marijuana, were able to correlate that as the THC concentration rose in the plasma or in the blood of the patients, the intraocular pressure went down in the eye. And this was significant, obviously, for obvious reasons, that potentially THC could be an effective therapy for glaucoma. But there were two major drawbacks related to this. One is when you smoke marijuana, the effect of THC is short-lived. It only lasts maybe 45 minutes to 90 minutes. And so if you wanted to manage your glaucoma, by smoking marijuana, you would have to smoke a lot of marijuana in order to manage your intraocular pressure, which of course is problematic. The second problem is actually a problem in the cardiovascular system. If you have to smoke that much cannabis, you're exposing your body to a lot of THC, probably more than what you need. And so what happens is that can lower blood pressure systemically in your body, and thereby lowering the blood pressure in your body can rob blood flow to the eye. And so the American Academy of Ophthalmology came out with a recommendation recommending do not smoke marijuana. to manage their glaucoma. But if someone could get the THC directly into the eye, that may be the best way to go. And so initially, people tried to develop THC eye drops in order to manage glaucoma. But THC, when you isolate it and purify it, is very what we call lipid soluble or fat soluble. And it almost looks like a drop of cooking oil. And so it doesn't get into the eye effectively. The University of Mississippi developed a method where they could bioengineer the molecule to make it more water soluble. And we have shown data in our presentations and also in publications to show that a much greater amount by using a prodrug to get the molecule into the eye, we're able to deliver orders of magnitude more THC into the eye than trying just to use a purified THC eye drop. Once the prodrug gets into the eye, there are natural chemicals in the body called esterases. These are natural enzymes that are in everybody's body. And what those esterases do is modify the prodrug, modify it to the point that it is turned into or metabolized into THC. The other aspect of this that's very important is that outside of the human brain, the human eye probably has one of the highest densities of cannabinoid receptors in the body. And interestingly enough, those receptors are located on organs that can modulate or regulate intraocular pressure. And so when THC binds those receptors, it results in a number of physiologic actions that as pharmaceutical scientists, we call the mechanism of action. And basically, one of the mechanisms of action is that when cannabinoid receptors are activated by the THC derived from the prodrug, it enhances drainage of fluid from the eye. And by doing that, it lowers the intraocular pressure. Which thereby makes it more effective as a drug for the treatment of glaucoma. The other facet of this that makes it an effective therapy in the eye is that cannabinoids as a general class of drug have been studied in multiple species of animals and have been shown to be neuroprotective. In other words, the retinal ganglion cells, these are the cells that make up the optic nerve and that die off when the pressure gets too high in the eye. Cannabinoids have been shown to be directly neuroprotective of these nerve cells. And this is why cannabinoids have been studied in epilepsy, in multiple sclerosis, in Parkinson's disease, because of this neuroprotective characteristic. So by using a cannabinoid therapy for glaucoma, you have the ability to affect the course of the disease in two major ways. You can lower the intraocular pressure to relieve the strain on the cells that make up the optic nerve, and these drugs, this class of drugs, especially THC that we have shown, is also directly neuroprotective to the optic nerve. So it's really, for lack of a better term, a double benefit of using a cannabinoid in the eye in the quest to make a better first-in-class drug for the treatment of glaucoma.
Now, you mentioned that there is other treatments currently on the market for treating glaucoma. What are those treatments, and how does this projected compound compare to those?
So currently, we only have animal data for our compound. Mm-hmm. We are planning to go into human trials next year, but animal studies, particularly studies done in rabbits, are highly predictive of what occurs in humans. And in fact, whatever dose you use in a rabbit, that dose is the dose that you use in a human. It's directly transferable. And what we have found is historically the most prescribed drug class for glaucoma is a drug class called prostaglandins. And in rabbits, prostaglandins can lower intraocular pressure anywhere between 30 and 35%. The second most prescribed class of drug is a drug called a beta blocker. Timolol is probably the prototype for that drug. In rabbits, it lowered intraocular pressure anywhere from 20 to 25%. And in humans, we see the same thing, 20 to 25%. Now, when it comes to cannabinoids, we already have human data of what THC can do based off of smoking cannabis and the studies performed by the NIH. And in those studies, those researchers found that THC could lower intraocular pressure anywhere as low as 40% to as high as 65%, depending on what the baseline intraocular pressure was. In our rabbit studies using a glaucoma model in rabbits, we have found that our prodrug of THC on average was able to lower intraocular pressure about 45%, which while it's not being conducted in a human, is consistent with historical comparables that shows that what happens in the rabbit is a pretty good predictor of what happens in humans. And so we've by virtue of history, where the human data was obtained first, usually it's the other way around. You do the animals first, then get human data. But because we have this history to draw on, we're able to at least say, based on the data we have to date, that our animal data does match up with the human experience from previous years. Now, naturally, we have to go through human clinical trials, and as I mentioned before, those are scheduled to begin next year.
Amazing. Now, you mentioned that the eye has multiple cannabinoid receptors. Are these CB1 receptors, CB2 receptors, or I guess what kind are they?
Well, not only you, but not, humans, dogs, cats, other types of animals, all of those species have cannabinoid receptors. And I apologize if I get a little geeky for a second, but from an evolutionary perspective, it's been estimated based on genetic data that these cannabinoid receptors go back hundreds of millions of years in evolution. So in understanding that, you have to scratch your head a little bit and say, there must be, going back to survival of the most adaptable or survival of the fittest, there must be some sort of survival benefit afforded by having cannabinoid receptors, given the fact that they have survived the millions of years of evolutionary development, and the fact that it's carried in so many species. So clearly, cannabinoid receptors serve an important function. What is a shame is because of the stigma that was associated with marijuana and the fact that it was illegal, and research was, because of the illegality, research was curtailed for many, many years and is only now trying to catch up, given the more liberal view of cannabis in general and the promising scientific data. It's just a shame that so much time was wasted when it could have been used to get a better understanding exactly of what these cannabinoid receptors do. Do they work in concert? There are two recognized cannabinoid receptors. Sometimes based on the exposure of the cannabinoid, they work together. Sometimes based on the concentration differential of a cannabinoid, they can work in opposite directions. And so only now are we beginning to understand on an organ by organ, disease by disease basis what exactly the interplay is between stimulating these cannabinoid receptors or blocking these cannabinoid receptors and the impact that can have on disease.
Interesting. Now, it looks like you guys are doing some work with CBD as well as THC. Is that correct? And if so, tell us a little bit more about that.
Correct. Correct. And so while the THC based drug is a prodrug, so again, a prodrug is an inactive compound that's bioengineered to enhance delivery of the drug into the body. And once in the body, it's metabolized and activated to the active part of the compound. And in this case, it would be THC. The CBD drug, on the other hand, is an analog. And an analog is a drug, is a molecule that has intrinsic activity in and of itself and can be bioengineered to help get that molecule into the body in a more effective and in a more predictable way. And also an analog could get metabolized to an active, a different compound that is also active. In the case of our analog of CBD, the CBD analog we've been able to show in some studies has its own activity. And then ultimately, that analog of CBD gets metabolized to CBD. So the way we're looking at studying this analog is that, in essence, because the parent compound or the analog has its own intrinsic activity, this could almost be viewed as being a long-acting type of CBD. Whereas you get the activity from the analog, and then ultimately you get a second round of activity as it's metabolized to CBD. To date, we have done a number of studies, both in ocular side of indications and also in the analgesic side. We have studied this analog of CBD in something called chemotherapy-induced peripheral neuropathy. Peripheral neuropathy is a very painful condition where the nerves become inflamed because of exposure to chemotherapeutic agent. Cisplatin, that's often used sometimes for breast cancer and lung cancer and some cancers of the bowel, is notorious for having this side effect. And of course, developing CIPN or chemotherapy-induced peripheral neuropathy is not a good thing, of course, from the basic aspect of developing a pain syndrome, but also is one of the leading causes of why patients have to stop chemo early. And of course, if you stop your chemo early, it can have a negative impact on your ability to clear the cancer. So having a substance available that can help patients tolerate chemo and mitigate this pain syndrome is very important, not only on the quality of life for the patient, but also to enhance their ability to rid their body of the cancer. And so we developed, in conjunction with our research colleagues at the University of Mississippi, we developed a research protocol where this chemotherapy-induced peripheral neuropathy, or CIPN, was induced in animals. And what we did was examine the ability of this analog of CBD to mitigate the pain that these animals were suffering. And what the long story short, we were able to show that our analog of CBD had analgesic capability comparable to morphine. And of course, morphine is probably just the basis of how morphine works, one of the most powerful analgesics out there, but of course, morphine is an opioid. And obviously, probably the vast majority of your listeners are aware of the opioid crisis currently going on in this country, where even short-term exposure to opioids can sometimes lead to addictive behavior, and then you have a problem of an opioid addiction. So what we are doing in conjunction with our research colleagues at Mississippi, we are examining the potential use of this analog of CBD as an alternative analgesic to opioids. A second part of that experiment that we did with the animals was a situation whereby the animals became addicted to a drug called OxyContin. And we were able to get the animals off of OxyContin and ultimately off of all meds by substituting our analog of CBD for the OxyContin. And by virtue of that, in the first experiment, we showed the analog of CBD had the ability to mitigate pain comparable to morphine. In the second part of the experiment, we were able to show that for animals that had an addiction to an opioid, we were able to help get them off of opioids by using this analog of CBD. And then I'm sorry, go ahead.
Just one quick question. How do you determine an animal is addicted to a substance?
So this is a standard procedure whereby animals can hit a bar in their cage, where by hitting the bar, it delivers a dose of opioid, and just like with people, in a very eerie way, some of the animals get to the point where they forsake food. They rather hit the bar and have the opioid delivered to them, even to the point rather than eating. And of course, you see this, the human equivalent of this, in a lot of patients that are addicted to fentanyl and to other powerful opioid agonists, where in some cases they're emaciated. All they care about is getting a dose of opioid to the point of forsaking spending money on food. They rather have that high rather than food maintenance. So it's really the same thing, but we were able, by using the cannabinoid, to get the animals to the point where they would actually eat and not keep hitting that bar for the delivery of the opioid.
Now, what are your thoughts on the addiction potential of THC?
This historically, this is a very mixed picture out there. And when people talk about addiction, there are really two major types of addiction. There's physiologic addiction, and obviously you see this a lot in opioids, where when you detox opioids or alcohol, for that matter, you go through something called something related to the DTs. You can go through violent shaking chills. You can go through essentially a withdrawal syndrome. So that's more in line with drugs that are physiologically addictive. Mm-hmm. Then on the other side of the coin, there is behavioral addiction. And the classic example you see is people that smoke a lot. There are addictive times, for lack of a better term, addictive times during the day. For instance, if they have a cup of coffee, you'll see people light up a cigarette. Or if after a meal, they'll light up a cigarette. It's not necessarily a physical addiction, but they've just become adapted. That behaviorally, that's part of their daily routine of when they will smoke.
Sure.
So the picture, how this relates to THC is historically it was believed that THC did not necessarily have a physical addiction related to it, but it could potentially have a behavioral addiction related to it, where people, their body doesn't necessarily crave it, but people believe that they need it. Uh, Now, some of that view is changing, and I think as far as addiction medicine is concerned, this is an area that's probably evolving as we have greater understanding of these receptor interactions. But I would say, excuse me, I would say that the physiological addiction of and I'm going to sound like everybody's mother or grandmother here, probably relates more to the old saying, everything in moderation. Excuse me. Uh, if you use it for a specific purpose to meet a medical need and you're not overdoing it, the potential for a physiological addiction is greatly reduced. However, if you're using it to the point of abuse, then whether it's a physiologic addiction or a behavioral addiction, if you're using it to the point of abuse, there probably is a risk of addiction, being either one of behavioral or physiological. And I think the area within the NIH, area called NIDA, the National Institute of Drug Abuse, I think they are exploring understanding this relationship. There's a lot of grant money that's being used from the NIH to get a better handle on this as the government anticipates more legalization, a wider breadth of legalization, and of course, from the medicinal point of view, more drugs getting approved that have a cannabinoid basis. So I would say everything in moderation, and this is an evolving story, and we'll see how ultimately how this all pans out.
Now, you mentioned earlier that THC has neuroprotective properties. It was long thought that THC may cause short term memory loss and dementia. What is the current understanding on that?
Well, I think this goes back to something, and I apologize, but given the question, I may have to get a little geeky here. Uh, this goes back not only THC, but all cannabinoids have something which pharmacologists call a biphasic response,
Hmm.
which basically says in low quantities, you will get one set of physiologic response and activity. But when you go higher in drug exposure, higher in concentration, you can actually get a paradoxical or the opposite response of what you wanted. The classic, since you brought up THC, the classic response is when you're exposed to low doses of THC, you get a feeling of well-being. You get a feeling of less anxiety. It almost has an antidepressive feeling. You're more relaxed. You may be able to sleep. It may have an ability to lift the anxiety enough that you're able to sleep at night. Now, if you keep increasing the exposure of THC, you end up with this biphasic response. And in extreme exposure to THC, using very high doses of exposure, you get almost the opposite response. You get anxiety. You get, not exactly a medical term, but you get something like jitters. You feel uncomfortable. You have insomnia. You can't get to sleep. And so it goes back to what we were just talking about as far as addiction and doing things in moderation. And I think this is why the government is taking a very hard look on the plant side of things. Back, even 15 years ago, the average THC concentration in plants was 6 to 8%.
Right.
And now you have people growing cannabis that is very, you're looking at anywhere from 20 to 30% THC. And, there can be a misconception out there among consumers that, well, if I felt this much better on 5% THC, imagine what I would feel on 25% THC. Well, cannabinoids, both THC and CBD, don't necessarily work like that. And so it would be important, and I think this is why the government is getting more involved, at looking at different, understanding the physiological, what happens to your body response to a low dose versus a high dose of a cannabinoid in that respect. So I would say, going back to your original question, THC taken responsibly, taken in a responsible dose should be okay. But if you kind of get sidetracked and if you're of the mindset that taking a lot of THC has got to be better than taking just a lower amount, it really is. I think people forget these compounds are just like any other drug out there. I would say maybe not a majority, but a significant percentage of our drugs out there are derived from plants. Tamoxifen for breast cancer, digoxin for heart arrhythmias, theophylline for asthma. These are all drugs that were originally derived from plants. Even aspirin. Aspirin was derived from the willow tree. I think people, in part of understanding and separating the stigma from the hype, really need to look at these cannabinoid molecules as these are just another group of potential therapeutics derived from a plant. And if you go and look, let's say, at aspirin derived from a plant, and if you take aspirin in the prescribed amount, aspirin is pretty safe. But if you go overboard and take too much aspirin, you can develop gastrointestinal bleeding. You can develop ringing in the ears, and a host of other side effects. So these compounds derived from the plant for cannabinoids are really just like any other drug. Taken in the right amount, very, very effective. But if too much of a good thing is bad. And so you need to make sure that you're staying within a decent dose exposure so you're not putting yourself at risk.
Now, that brings up another really interesting conversation because, as some of our listeners might know, Epidiolex is a really high dose version of CBD. And there's thought in the larger cannabis community that you can decrease that dosing if you're able to use a full spectrum cannabinoid plant tincture where you are basically having access to the entourage effect. What are your thoughts on that one?
Well, I would say this, and again, I don't want to speak for another drug company, but what I can do is comment on what was released by the FDA, and basically any drug that you take into your body, that drug can interact with food. That drug can interact with other drugs. CBD, whether you derive it from the plant, whether you get it from an edible, or whether you get it from a drug like Epidiolex, has the potential, because you're ingesting it orally and it goes through the gastrointestinal tract, has the potential to interact with the cells in the liver. The liver is the detoxifying organ in the body and breaks down drugs. So it can interact not only at the time it's being broken down with the liver cells, but potentially those liver cells are impacted by other drug medications you may be taking and by other drugs. So you have to understand a number of principles when taking a drug. Epidiolex showed that in the case of controlling seizures or epilepsy, the dose exposure was about 20 in the area of 20 micrograms per kilogram of drug per kilogram in order to control seizures. That concentration in your body, if you're taking other drugs, could get elevated, or if you're taking a different set of drugs, it could be lowered. So it really goes back to go back to the basic advice given by the FDA. If you're taking any type of cannabinoid by any type of route of administration with the focus on CBD, the safest thing to do is to inform your doctor. Let your doctor know so that the doctor can weigh the risks and the benefits based on an understanding of what other medications you're taking. I think that's probably the soundest advice. Always let your doctor know, and even so far as not just prescription drugs that you may be taking, but also let's say natural or herbal drugs, say John's wort, echinacea. Even though these are not prescriptive drugs, they can affect the metabolism of other things that you may be taking that are prescriptive. So I would say overall, always let your doctor know what you're taking so that your doctor can figure out what the risks are based on your overall health and other medicines that you're taking.
So obviously, there's a lot of hype about CBD right now. There are quite a few people that are taking it for just general wellness and energy and things like that. What are your thoughts on that?
Well, um, I look at CBD from the perspective of CBD as a drug.
Hmm.
It's not a natural body constituent, although there are receptors in your body that can bind it. But I should also add, your body naturally makes to its own cannabinoids,
Hmm.
and hence we call the receptor system throughout your body the endocannabinoid system. So your body does make two cannabinoid-like molecules in and of itself, and that's what that endocannabinoid system was designed to interact with. For CBD, again, I think it goes back to the following. And again, this is based off of FDA analysis and also analyses performed by the NIH. If you are taking a CBD supplement, now if you're taking a prescriptive CBD medicine, you at least know that the drug company had to meet manufacturing standards in order to make it, and that also what it says on the label. If the label says there's 20 milligrams of CBD in the in that pill, you can be 99% positive there's exactly 20 milligrams of CBD in that pill, and it's purified, and the excipients that are used to make the pill have been studied in trials. On the other hand, if you're relying on CBD from a non-pharmaceutical source, then things that you need to be very careful of is to look on the label. What is the amount of CBD that they are saying that's in the product? Where was that CBD derived from? If it was derived from the plant, and there are a number of companies that let's say derive CBD from hemp, are they reputable companies? Have they ever been cited? Are there other things within that formulation that you're taking the CBD in that may not be healthy for you? such as pesticides or heavy metals that are used in the process of extracting the cannabinoid from the plant. How reliable is the amount of CBD in that product? One batch of product may actually have 20 milligrams of CBD in it. The other batch may only have 12. But they made it the same way, but maybe there wasn't that good manufacturing process. So I would caution your listeners out there, if they are getting CBD from a non-pharmaceutical source, to be very, very careful as to the reliability of that source. You can check the FDA website, both the FDA and the DEA website, to see if any of the producers of a compound that you may be taking, if they have been cited in the past for manufacturing problems. Now, the fact that they're not on the list doesn't mean they may not have been screened yet by the DEA or the FDA, but it's a good first round screen to check. But we want to make sure everybody that is taking CBD or any drug product really, is doing it in the safest manner possible.
As an executive in this industry, do you feel that there's room for both pharmaceutical grade cannabinoids as well as medicinal grade? Or what are your thoughts on how the industry is evolving? Because it is evolving as we speak.
Sure. I would say overall, I would say the following. There will always be people that prefer the source of their cannabinoids from a plant as opposed to, let's say, a synthetic version of the cannabinoid. That's number one. Number two, I would add, though, that the receptors in your body cannot distinguish a THC from a plant versus THC made in the lab. Mm-hmm. Those molecules are the same, and the receptors cannot tell the difference. Number three, I think we're seeing an era now where there's a new player as far as the source of material. So you originally had cannabinoids derived from a plant. Then you had cannabinoids chemically synthesized in a lab, which dramatically cuts down on the cost of making the cannabinoids. And you can make a more cohesive kind of product through chemical synthesis. And then the third type of manufacturing now that we're seeing the advent of is biosynthetic synthesis, whereby the genetic material from the plant is inserted either into yeast or bacteria and are fermented to make tremendous amounts of cannabinoids at a fraction of the cost of what it would take to actually grow a plant and make them. So these are the three areas that are under development now. And I would say probably one force of nature in all of this that people don't really talk about that much is the insurance industry. Ultimately, as I mentioned before, CBD from a plant or CBD manufactured, your body can't tell the difference, but your accountant may be able to tell the difference because if to get CBD derived from plants, to get a month's worth of that, can maybe run a couple of $100. Right. But if you have a prescription for CBD, once something is approved by the FDA, your insurance company has to cover it. So if you have health insurance, instead of spending a few $100s a month, it might be a copay of anywhere from $5 to $15 a month. Right. Which is a dramatic cost savings. So there are a lot of different forces at play here. Ultimately, it ends up in the consumer's consumer's decision as to what's best for them. And probably the most reliable thing to say on all of this is to stay tuned. We have to see how this all plays out. But ultimately, as I said before, there are a certain group of people out there that are wedded to the plant, that think the plant is best for them. And, there's nothing wrong with that as long as they're sure that whatever they're getting from the plant is reliably manufactured and is safe for them.
Amazing conversation. Thank you for sharing so much of your wisdom and spending your morning with us. I look forward to perhaps future conversations about the studies and what they show, and maybe we can develop this conversation further.
Terrific. And thank you so much, time, and thank you to your listeners. Thank you.
Bye-bye.
Bye-bye now.